shall now discuss on gustation, the sense of taste. Have you ever wondered how we are getting different tastes for different foods we intake? So today we are going into detail the basic mechanism behind how we are getting the taste. Taste, like olfaction, it's a chemical sense. And as we all know, there are five basic tastes, five primary tastes, which is sour, sweet, bitter, salty, and umami. And what is this umami? It tastes like something meaty or savory and was first discovered by a Japanese scientist from a university in Tokyo. And umami, it is believed to arise from taste receptors that are activated by monosodium glutamate or MSG. That is what we commonly call as Ajinomoto. It is present naturally in some food substances and can also be added as flavors to enhance the flavors. And all other flavors like chocolate, pepper, coffee, etc. are all combinations of these primary tastes along with olfactory and tactile sensations. And odors from our food pass upward from our mouth to the nasal cavity where it stimulates our olfactory system. And since olfactory system is much more sensitive than our gustatory system, a given concentration of our food can activate our olfactory system 1,000 times more than it could activate the gustatory system. And have you ever felt that when you're having cold or allergies, you cannot feel the actual taste of a food? It is not because we are having gustatory system blocked, but the actual system which is blocked is our olfactory system. Let's go into the anatomy of the taste buds and papilla. The receptors for our sensation of taste, it lies on the taste buds. And nearly there are 10,000 taste buds on the tongue of a young adult. And apart from this, there are also taste buds on the epiglottis, on the throat, on the pharynx, etc. And the number of taste buds declines or decreases when we get aged. That is why when we hear our parents or grandparents saying that there is no taste for the food. And when we go into the structure of each taste buds, it consists of three types of epithelial cells called supporting cells, gustatory receptor cells, and basal cells. This is the supporting cell, this one is the gustatory cell, and this is the basal cell. Supporting cells, it surrounds about 50 gustatory receptor cells in each taste bud. And next comes the gustatory cell. This gustatory cell possesses a long microvilli-like structure from each gustatory receptor cells and opens to the external surface through the taste pores. And the next comes the basal cell. They are stem cells, which give rise to supporting cells, which in turn then give rise to the gustatory receptor cells. And these stem cells are located on the periphery of the taste buds and are connected to the sensory epithelial cell or connective tissue layer. And this is the structure which I said now. Each taste buds on a papilla. The structure of a single taste buds, and these are taste buds arranged in a row with gustatory cells, supporting cells, basal cells, connective tissue layer, taste pore, the opening, and the gustatory hair cells, which are microvilli-like structures. And at their base, the base of each taste buds, the gustatory receptor cells synapse with the dendrites of first order sensory neurons, which forms the first part of the gustatory pathway. And next, the dendrites of each of these first order neurons branch profusely and contact with many gustatory receptor cells in several taste buds. And next, time for a question. Why doesn't take taste sensors on the tongue too long to recover from being burned by a hot coffee or tea? The reason is that each gustatory receptors have a lifespan of only 10 days. And we could now look on to the structure of or different types of taste buds. taste buds are found on elevations of our tongue called papillae, and the function of this papillae is to increase the surface area and provide a rough texture to the surface of our tongue. And the three types of papillae include circumvallate papillae, fungiform papillae, and foliate papillae. And first one, the circumvallate papillae. You can see an inverted V-shaped round structures on this part of our tongue, and this forms the circumvallate papillae, which is 12 in numbers, and each of these papillae contains around 100 to 300 taste buds. And next comes the fungiform papillae. As the name indicates, they are mushroom-shaped papillae arranged throughout the surface of our tongue. You can see some small dots here. These are the mushroom-shaped papillae, and this mushroom-shaped papillae contains around 5 taste buds in one papillae. And next comes the foliate papillae. These are small trenches. See here, these are small trenches on the lateral margins of our tongue, and the number of taste buds decreases in the childhood age itself. And next comes the filiform papillae. As the name indicates, they are thread-like structures found throughout the surface of the tongue. See, these pointed thread-like structures present throughout the surface of our tongue is called filiform papillae, and their function is to give a tactile sensation. They are tactile receptors and not taste receptors. And their function is to increase the friction between tongue and the food and to make it easier for the tongue to move the food into the oral cavity. And next comes the actual part, the physiology of gustation. Chemicals that stimulate our taste through the gustatory receptor cells are called tastens, or the chemical molecules which gives the taste. It is called as tastens. And once the tastent is mixed with the saliva, it comes in contact with the primary gustatory receptor cell, plasma membrane of the gustatory receptor cell. And this results in the generation of a receptor potential, leading to the exocytosis of synaptic neurotransmitters stored in the synaptic vesicles. And these liberated neurotransmitter molecules then trigger nerve impulses in the first order sensory neurons that synapse with the gustatory receptor cell. And the receptor potential arises differently for different tastings. That is, if you are taking a salty food, the Na plus ions enter the gustatory receptor cells via sodium channels in the plasma membrane. And as the accumulation of Na plus ion increases, it results in depolarization, leading to the release of neurotransmitters. As like this, if you are taking a sour tastant, H plus ions may flow into the gustatory receptor cells via H plus channels. Anyhow, whether it is Na plus ions or H plus ions, it results in the opening and closing of several types of ion channels, resulting in depolarization and liberation of neurotransmitters. And there are other tastes like sweet, umami, and bitter. What happens to these tastents? These tastents do not directly enter into the plasma membrane of the gustatory receptor cell. Rather, they bind with certain receptors on the plasma membrane which are coupled to G protein. That is, they are G protein coupled receptors. And through these ion channels in the plasma receptors on the plasma membrane, they activate the G protein, which in turn activates many second messengers inside the gustatory receptor cells. Anyhow, all these result in the same release of neurotransmitters, but in a different way for different tastings. And let's think, if all the tastings cause the release of neurotransmitters from many gustatory receptor cells, why do foods taste different? The answer is that the different tastings arise from the activation of different groups of taste neurons. That is, although each individual gustatory receptor cells respond to the membrane responds to more than one of the 5 primary tastes, it may respond more strongly to some taste than to others. And there is something called taste thresholds and adaptations. That is, complete adaptation to a specific taste. It takes around 1 to 5 minutes. That is, it takes around 1 to 5 minutes for us to get a specific taste. And this is due to this taste adaptation is due to the changes in taste receptor cells or olfactory receptor cells or even in the neurons of the gustatory pathway in the central nervous system. And the threshold taste for each taste, it varies. For example, for bitter, such as quinine, bitter taste such as quinine, we need only a very little amount of this quinine tastings to get the bitter taste. It is actually an adaptation for us to avoid the bitterness or some poisonous substances. And the threshold for sour, such as lemon, it needs a somewhat little more higher amount. And this is the visualization of the gustatory pathway. Let's go into it. The gustatory pathway. See here, there are 3 cranial nerves involved in the gustatory pathway. They are, the first one, the facial nerve. It receives taste buds. It receives the impulses from the taste buds in the anterior 2/3 portion of our tongue. This is also the 7th cranial nerve. And next is the glossopharyngeal nerve or 9th cranial nerve. It receives the sensations from the posterior part of our tongue to the brain. And the last one is the vagus nerve or 10th cranial nerve, which receives or propagates impulses from the epiglottis and throat region of our tongue. And these 3 cranial nerves together propagate the impulses to the gustatory nucleus in the medullary region or medulla oblongata. And from here, the impulses are propelled through into the hypothalamic region to the thalamus and then to the primary gustatory area of our cerebral cortex. That is, once again, from the taste buds, nerve impulses propagate along these cranial nerves, the 3 cranial nerves, to the gustatory nuclei in our medulla oblongata, from there to the limbic system, hypothalamus, and then into the thalamus. And from the thalamus, the primary gustatory area in the parietal lobe of our cerebral cortex, and this gives rise to a conscious perception of taste. So, have a happy meal.